So, welcome back to the NPTEL lecture series on bioelectricity. So, let us do a bit of a recap. So, what we did, we started with the stretch reflex arc circuit and we talked about muscle spindle, and we talked about how the muscle spindle can anticipate the change in length of a muscle, and it is innervated by the sensory neuron, and the sensory neuron takes the message to the spinal cord, and in the spinal cord this message is transmitted to the motor neuron through an interneuron, and then the motor neuron brings back the message telling the muscle to come back to its original position, regaining its original length, and simultaneously there is another set of motor neuron which are the gamma motor neuron, which tells the muscle spindle to come back to its original shape. So, in this process today we will be briefly discussing how the signal from the muscle spindle is transmitted to the sensory neuron, okay, and how that could be measured. So, coming back to the slides. So, this is lecture 15. So, essentially this is the situation. Here you have the muscle with all the myotubes and everything, likewise striations and all those things you see. Okay. In between you are having these muscle spindles sitting there, and these muscle spindles are innervated by, let me use a contrasting color, something like this. They are innervated by the sensory neuron ending. So, our pertinent question is how this signal from here is being transmitted from this muscle spindle to sensory neuron, and what we know about it in terms of its electrical signature. To sensory neuron. Okay. How this electrical signal transfer is taking place. So, it is believed that there may be a series of sodium channels which are either present in the muscle spindle or they are present in the sensory neuron. These channels are, again, this is all currently under speculation. These are believed to be mechanosensitive ion channels. So, these kind of sodium channels are considered as mechanosensitive ion channels. Okay. What we meant by mechanosensitive ion channel, as the name itself indicates, because of mechanical stimulus the ion gates open up. So, it has a very unique way of opening up. So, this is a classic case of mechanotransduction, or in other words mechanical energy leading to the generation of an electrical energy, or mechanical energy is converting into electrical energy, whichever way you want to put it. So, essentially this is the first level of energy transduction which is taking place, and so essentially what is happening is that this muscle spindle is getting a mechanical force, and this or this is not yet clear, opens up a series of sodium or cationic channels. It may be even calcium, which is not really known, and this leads to a generation of an action potential. This is how far we know about how the interfusal fibers are communicating the signals to the sensory neurons. things are under intense study, but still we have a long way to go before we really understand how this first level of information transfer is being executed. Okay. So, this is the first level. What is the second level? Second level, so this is, here is the train of action potential which is traveling through, fine. Likewise. Now that the level, so let us put it at level 1. Level 1. What is happening in level 2? Coming back to level 2. So, a signal has arrived from the sensory neuron. This is sensory neuron, and through the interneurons, this signal is getting split up into two different components. Okay. So, one component is going for once again. So, this is the train of action potential which is traveling. Okay. Now, here this signal is getting split up into two components. One component, say I am representing by green, the other component I am representing by red. So, one component of this signal, if this signal I represent by some say function, say f x is equal to s, then here we will have two signal f x 1 and f x 2, fine, or you can represent it by any way. Okay. And essentially signal s, so f x is equal to f x 1 plus f x 2. This is how the mathematically this could be defined. Now, from here this signal is transmitted to two different kind of neurons. One is a smaller one which is called a gamma motor neuron, and the other one is the bigger one which is called an alpha motor neuron. Okay. Just for the understanding sake, and here you have all the synapses and everything, and fine. So, this is alpha motor neuron. Now, this alpha motor neuron is the one which is responsible for contraction of the extrafusal fiber or the bulk of the muscle, and the gamma is the one which ensures that the muscle spindle comes back to its original shape. So, this is, if this is level 2, then at this stage there are 3 levels which are starting. From here we are initiating level 3. So, here there is the train of action potential which is traveling to ensure that the muscle spindle regain its original shape or length. Again, this part of the circuit, how the gamma motor neuron is executing this function is still not very clear. We know the end result, that is how it does, but the real mechanistic details are still not very clear. Instead, the other part of the circuit where the alpha motor neuron is regulating the muscle length is much more clear. Okay. So, now what we will do, we will talk in bit of a bigger details or a greater details about this particular part of the circuit, where the alpha motor neuron is communicating its information to the extrafusal fiber, and this it does at neuromuscular junction. Okay. So, this is now where we are moving into the level 4, which is of this circuit, level 4, which is neuromuscular junction. In short, It is also called NMJ. So, neuromuscular junction, pretty much all of the vertebrates are regulated by a specific kind of neurotransmitter called acetylcholine. Barring aside some of the neuromuscular junctions which are present in the Drosophila, which are driven by glutamate kind of neurotransmitters. So, we will not take those exceptional case into account, we will only talk about the acetylcholine based nerve muscle junction, where your alpha motor neurons are coming, and let us represent the alpha motor neuron by red. These are the huge neurons, and they are the one which are coming and innervating the muscle. Here you have the muscle. Okay. So, and the input it is receiving from the interneuron or directly from the sensory neuron. So, here is the input which is arriving at its different terminals. These are the dendritic terminals where the input it is receiving all the inputs. Okay. So, the direction of flow of electrical stimulus is like this. So, there is a form of spatial and temporal summation of information. Spatial in the sense, because if you look at this image out here, if you look at this whole image, signals are coming from different location in a space. It is coming from here, so it is coming from here, it is coming from here, it is coming from here, it is coming from here. Okay. So, that is why it is spatial and it is also temporal, because the signal may come at time t1, time t2, time t3, time t4, and one signal may overlap over another signal. So, say for example, like this, then another signal coming like this, then another signal coming like this, another signal coming like this. Okay. If this one is t1, this is t2, this is t3, this is t4. So, this is also called temporal. So, in other words, electrical signals which are arriving at the motor neuron are electrical signal, or you can call it or train of action potentials are spatio-temporal. They are spatial as well as temporal in nature. And this motor neuron, what it does, it is just like a computer out here. Okay? It integrate all this signal, and this integrated signal is now the master signal which starts to travel down like this along the axon, without allowing any kind of back propagation of information. Okay. In this direction it is moving. So, what essentially is moving is a train of action potential which is coded with the information that how much length the muscle has to come back. Okay. Now, what happens here? So, at this stage there are two things which I wish to bring to your notice. First of all, these neurons are myelinated, and if you look at these neurons, they are so if this is your motor neuron, and this is how the process is. Okay, so now okay. Right. And here you have the nucleus. So, these are all myelinated structures. Myelinated in the sense they are something like this. Okay. So, these are very similar to, you have seen the wires on the walls. Okay. They are very similar to the wires on the walls. You cannot have a naked wire in the wall, on the walls, because if you have a naked wire, then if you touch it, you will get a shock. So, in order to prevent that, the same thing holds true for these neurons, because they are conducting electrical impulses. So, the current should not be lost, the information should not be lost. So, that is why it is put inside an insulator, and these insulating cells are, in the case of motor neuron, when the motor neuron, so as long as this motor neuron is inside the spinal cord, it is myelinated, or the insulation is done with a specific kind of cells called oligodendrocytes, and when it is outside the central nervous system, it is insulated by another kind of neuron, another kind of glial cells, which are the supporting cells of the nervous system. Those are called Schwann cells. Okay. We will come in depth on those, we will be talking about some of the diseases and how to really handle it, but at this time just remember, these green covering what I have drawn, they do not allow the electrical signal to be lost from the axon to the another one. There should not be any short circuit. Okay. So, these are your, they could be Schwann cells or even oligodendrocytes. They ensure insulating covering of the axon. Right. So, now this zone where I am circling now is called the axon hillock, and these small gaps, what you see out here, what I am labeling now, are called nodes of Ranvier, and at the nodes of Ranvier you have a large population of sodium channels. They are concentrated out here like this. What you see in green are high population of voltage gated sodium channels, and the current which is now it is obtaining from all the sources and it is doing the summation. This current actually hops like this, it is called saltatory conduction mode, okay. Like this. Saltatory conduction mode. Okay. So, at this stage what is happening? Now, once you are at the zone where it is in contact with, okay. So, this is the zone where this whole structure is in contact with the muscle. Okay. So, now at this stage, these electrical signals had to lead is to translate in information to the muscle, and it does at these junctions, which are very, very specialized junctions. It is these are the junctions where this information is being transmitted and how it does so. Now, what we will do, we have talked about the movement of the electrical impulse out here. What is happening out here? So, we will magnify these images of neuromuscular junction, and we will talk about them in the next slide. So, let us redraw the neuromuscular junction now. So, if I to redraw the neuromuscular junction, it will be something like this. Something like very similar to the way I am drawing it, and we will do the nomenclature very soon. So, this is the neuronal terminal, and here you have the muscle. Okay. So, at this stage, what you see out here essentially, these are filled with something like vesicles or small packets of neurotransmitters. These neurotransmitters are the molecules which translate the electrical impulses into chemical impulses. I will come to that. Okay. So, these are the small vesicles. These are filled with, in this situation when we are discussing about neuromuscular junction in mammals and everything, they are filled with a chemical called acetylcholine, okay, like this. And these neurotransmitters are actually synthesized by the neuron, and they are transmitted at these different terminals. And the byproduct of it is actually also carried back to the cell body and again resynthesized and brought it back. So, there are 5 different steps what is happening out here. Okay. The first step is, so let us start enumerating the steps now. So, the step 1 of this game is step 1, then arrival of action potential, arrival of, I am just putting P for potential, and which basically depolarizes the synaptic knobs. So, just let me introduce 2 more terminologies. The one which is sending the signal, it is called the presynaptic, and the one which is receiving the signal is called postsynaptic. In this situation, the presynaptic membrane is the neuron and the postsynaptic membrane is the muscle. So, this is postsynaptic and this is presynaptic. Okay. And these terminals, these are called synaptic knobs, and this small gap, physical gap, this is called nerve muscle synapse. So, as a step 1, with the arrival of action potential, it depolarizes the synaptic knobs. So, what essentially that mean? That mean, coming back to the slide, that essentially mean that the step, what is happening is that, so if this is the, and if this is the muscle, sorry, this is the muscle. So, out here when the action potential arrives, this leads to the entry of, they are depolarizing, in other words, they are trying to make the membrane much more positive, which was at minus 90 or minus 80. Okay. So, now with the entry of the calcium, what will happen, there will be this membrane potential will start to depolarize from the negatively polarized state of minus 80 to minus 90. With the entry of the calcium, it will become more positive. As it becomes more positive, the next thing what will happen is this. So, coming back, so there is an entry of calcium, which is the step 1. This followed by this calcium goes, coming back to this. So, with the entry of the calcium, this calcium goes and bind to these synaptic vesicles. And as soon as it binds to these synaptic vesicles, they open up. They started secreting the neurotransmitter in this zone. So, these are the acetylcholine which are being secreted by the nerve terminal. This acetylcholine immediately goes and binds to the postsynaptic membrane. It's coming back to the postsynaptic. On the postsynaptic, there are receptors for the acetylcholine, and they bind on top of them. As soon as they bind on top of these postsynaptic membranes, this opens up a series of cation channels. So, what is happening? Visualize this slide. So these are the terminals. Okay, and so this is where the synapses are, right? Like this. This is the muscle. So this muscle has a series of receptors which binds to acetylcholine. So the green one. Okay. Let's represent them by, let's pick up a color which is much more prominent, which would be seen out here, something like this. Okay. Let me make it thicker. Okay. So the dots what I am drawing now are the receptor for acetylcholine on the postsynaptic or on the muscle membrane. So as soon as the acetylcholine is released into the synaptic cleft or the space between presynaptic and postsynaptic membrane, that acetylcholine goes and binds to the postsynaptic membrane. Once it binds to the postsynaptic membrane, it leads to the influx of sodium into the muscle, and that sodium which is generated into the muscle leads to the generation of an action potential by the muscle. And this leads to a generation of action potential. So, if we have to put it in perspective, think of what all happened. Let us go back to the first slide where we started. Okay. So, this is how the circuit looks like. From muscle spindle, because of presence of mechanosensitive ion channels, the muscle spindle kind of stretches, and this mechanical signal is translated into electrical signal. Then this electrical signal travels through to the stage 2, to the interneurons. In the interneuron, the signal is divided into two parts. One goes to gamma, one goes to alpha. The gamma electrical information comes to the muscle spindle and gets translated it into mechanical signal to tell the muscle spindle to come back to its original position. Whereas, on the other hand, in the case of alpha, these electrical signals travel along the alpha motor neurons, as I was showing you out here, without getting lost. They travel out here, and after reaching here, they depolarize the membrane by allowing the entry of calcium into the cytoplasm. With the entry of calcium, the membrane got depolarized. Once the membrane got depolarized, the calcium in the meantime goes and binds to the synaptic knobs, which are filled with vessels of acetylcholine, and once the acetylcholine is released into the synaptic cleft or in the neuromuscular junction, that acetylcholine goes and bind on the postsynaptic receptors which are present on the postsynaptic membrane and leads to the entry of the sodium ions into the muscle, and then the muscle generates an action potential. So, electrical, chemical, again electrical, and then what we haven't discussed, how this action potential within the muscle leads to a generation of a muscle contraction. We will come to that, but before we come to that, now the way these are being studied is known. So, if you could put an electrode here and if you could put an electrode here, and if you stimulate it, if you stimulate the presynaptic membrane, and then you could make the recording from the postsynaptic membrane. So, you can put an electrode here like this, you can put an electrode like this. This is how all these studies over last 70, 80 years has been characterized. All the muscle contraction, all the in vivo studies, there are in vitro preparation where outside the body you have neuromuscular junctions, you have live animal preparation, there are whole series of, there are slice preparations where all these electrical measurements are being done, all the quantas are being released. As a matter of fact, studying the electrical properties of neuromuscular junction has opened up a wide window of understanding the synapse itself, because this is one of the very well accessible synapse where you really can see how the ultrastructure of synapse looks like, because all the synapses are not that big. This is huge. You really can see in any muscle of your body, you really can see a neuromuscular junction. So, if there is a cross section from an experimental animal, you actually can do a electron micrograph and everything, and you can really figure out what is happening. And this whole site, what you see here, is one of the hot site for pharmacological intervention. There are several diseases which are related to it. So, one of the thing what happens when acetylcholine is being secreted. So, acetylcholine binds, but very soon, very immediately, pretty much at the same time, the acetylcholine has to be degraded out. So, what has to be done essentially is, let me go to so the acetylcholine which is secreted is being degraded out as choline and acetate. Choline and acetate, and this choline is again taken up by the neuron in order to make new acetylcholine, okay, and acetate is further degraded, and this is being done by an enzyme called acetylcholine esterase. So, if you have a blocker for acetylcholine esterase, then the synaptic transmission will be disbalanced. There will not be any control route, because then they will keep the channels open and the individual muscle will start shaking, and there are several poisons which actually does that. They ensure that acetylcholine esterase does not break down, or something very similar to acetylcholine, or something which blocks to acetylcholine esterase, or something of that kind. Okay. So, this is another feedback mechanism by which it is being ensured at the synapse, the neurotransmitter which is translating the electrical signal or transferring the electrical signal from the neuron or from the presynaptic terminal to the postsynaptic terminal, in this situation the muscle, that neurotransmitter should have a very, very limited lifetime. If it is not so, then there will be problem. We will have serious issues if this biochemical pathway is not being regulated the way it is. Okay. There is another thing which is worth mentioning here, as I was trying to tell you, though this is not part of this, but just for your understanding sake, the way it works is, so all the, if I have to show you in 3 dimension like this, that will be easy for you to understand. So, all these neurotransmitters are actually synthesized out in the nucleus, and they are transported out into the nerve terminals, and their byproducts like choline or something, they are transported back out here. And this whole transport mechanism is very, very essential, because at times in several diseases, several neurodegenerative diseases, it has been observed that this transport phenomena is obstructed. So, this is called a trafficking. Those who are not from a very well versed biology background for them. So, this is what is the trafficking process, when neurotransmitters are being put in vesicles, come back and they adhere and form the synaptic knobs out here, the neuromuscular junction, and then they are being again taken back after they are being used. Okay. So, this is a continuous trafficking process, and this is called anterograde and retrograde transport, and retrograde transport of vesicles. As I have already told you that you can study them by putting an electrode out here and putting an electrode at the other terminals. If you see if you give a stimulus at E1, if this is E1 and this is E2, then just with a slight gap you will see a signal at electrode at E2. That gap is the time what it takes from here to travel to the postsynaptic membrane. Okay. And if you do not see such signal, it means they may be physically in close to each other, but there is no functional connection. These are acid tests of figuring out whether your motor neuron is sending a signal or not. But this takes us to another level of complexity, which I am going to dealt in the next class, that is